So today, we're going to talk about, in my talk, some of the cell biology of RanBP2 Nup358. Okay, so before we go there, I just want to have just a general discussion on the nuclear pore, viruses, and the innate immune response. And from there, we'll transition to how RanBP2 plays a role in each of these various cellular events. So I just wanted to first off just comment that human cells are divided into two major compartments, the nucleus and the cytoplasm. And if we were to zoom in here, the nucleus where DNA is stored and the cytoplasm where information from DNA is used to make proteins. Well, what divides these two compartments is the nuclear envelope, and molecules have to actually move between these various compartments. And the major way, and most of the time the only way that molecules can travel between the nucleus and the cytoplasm is through the nuclear pore complex. Okay, so if we take a closer look at the nuclear pore complex, and in particular, these filaments that are on the cytoplasmic side of the nuclear pore. So here's just a transvert, I don't know if you can see my mouse. Maybe I should make this into a pointer.
No, we can see it.
You could see the mouse. So here we're just looking at transverse section of the nuclear pore, but if we were to look at it using what is called micrographs. You can see here's a membrane and several pores with a basket and filaments. And then if we were to sort of rotate the nuclear pore, so we're looking at it face on, so this would be on the surface of the nuclear envelope, you can see because we cut the pore in half, you only see half of it and you see four filaments. And when we look at micrographs, we can see that the entire pore has eight of these filaments. And this has been seen through a variety of different microscopic types of techniques. Actually, here's a nice picture of the basket that's on the inside and of the filaments that are on the outside of the nuclear pore. So the nuclear pore, being the main conduit between these two subcellular spaces, is a hub for viruses and the innate immune system. And so why is that? Well, when viruses infect cells, actually cells can sense these, and signals that start off in the cytoplasm or in other cells, and those signals then have to traverse through the cytoplasm, they have to cross the nuclear pore so those signals can activate genes that are involved in the innate immune response pathway. And so the pore is kind of a necessary route through which these signals must traverse. And once those genes are activated, information in the genes is copied in mRNA molecules, and those mRNA molecules then have to exit again through the nuclear pore so that they are used in protein synthesis. So the mRNAs contain the information on how to make new proteins, and these might be antiviral proteins or secreted proteins that act as warning signals to nearby cells that this cell is infected. And some of those signals include cytokines, which many of you have likely heard from our clinical colleagues play an important role in mediating some of the symptoms of acute necrotizing encephalopathy. So the nuclear pore is not only used by the cells, but is actually accessed by the viruses. Many viruses actually have to enter the nucleus to complete their life cycle. And many of these viruses actually dock onto the pore. Some of them traverse the pore, and some viruses actually shed proteins that interact with the pore. But there seems to be a lot of connections between viruses and the pore itself. And these viruses, many of them are trying to get access to the nucleoplasm to complete their life cycle. And because of this sort of bottleneck, a figurative and actually quite literal bottleneck, the nuclear pore ends up being a battleground between viruses and the innate immune system. So any antiviral signals that are trying to cross through the pore, viruses attempt to um, circuit, basically short circuit, sorry, the system by inhibiting these antiviral signals before they can enter the pore. And even if the antiviral signals enter the pore and activate innate immune response genes, well, those genes produce mRNAs that then have to cross right back out of the pore. And in some cases, it appears that viruses try to mess around with the mRNAs for their own benefit and manipulate the innate immune system. At the same time, it's likely that cells monitor their nuclear pore to see if viruses are attached. And it's likely that when viruses do attach to the nuclear pore, this in itself may activate innate immune response genes. Likewise, mRNAs that are important for antiviral response may respond to altered pores that show tampering by viruses. Okay. So now that we've discussed the pore in general, viruses, and the innate immune response, we want to take a closer look at RanBP2. So RanBP2 is also known as Nup358. This is one of these quirks of cell biology that when genes are discovered independently by multiple groups, they end up with multiple names. I know that when I was first in contact with the ANE community, there was a lot of confusion about this. But essentially, RanBP2 and Nup358 are the same thing. It's just that they were named by independent groups. And so RanBP2 sits on the cytoplasmic face of the nuclear pore and is the main protein in these filaments. And if we zoom into these filaments, we can see that every single filament is actually composed of 5 RanBP2 protein molecules. Okay? And I'm just showing here on the right a single one of those molecules, and you can see that they're the RanBP2 protein, which is made from the RanBP2 gene, is divided up into chunks, which cell biologists and biochemists like to refer to as domains. So there's various domains that make up the entirety of RanBP2. And some key important domains is this N-terminal domain, where a lot of the ANE1 mutations reside. And you can see that this domain tends to bind to the nuclear pore, a part of the pore that forms these rings that are called the Y complex. So the N-terminal region binds there. And the ANE1 mutations tend to, at least the most common ones, tend to cluster on this top region of the N-terminal domain. Now, I have to say that there are other documented ANE mutations that are widely distributed across the gene that affect different types of the parts of the protein, but the ANE mutations, especially T585M, is present actually around here. Another important domain that I'm going to talk about is this sumo E3 ligase domain, and I'm just going to spend a couple of seconds here talking about what that is. So this is just the zoom up of this domain, and this E3 ligase domain actually forms a tight association with a couple of proteins. One of them is called RanGap1, and it not only binds to RanGap1, but it binds to a sumo ligated form of RanGap1. And on top of that, this E3 ligase domain binds to UBC9, which is also known as the E2 sumo ligase. And this is attached to sumo, but UBC9, unlike RanGap1, can do something tricky with its sumo. It actually transfers that sumo to other proteins that are nearby. And so this is known as sumolation, right? And so RanBP2, by virtue of the fact that it binds to UBC9, can actually promote the transfer of this sumo, which is a small protein in and of itself, to be attached to various protein substrates. And that modulates the activity of these protein substrates. Now, we don't understand what activates the sumolation event. It's likely triggered by things, but we have actually at this point really no information as to what triggers that. And it's likely that this type of sumolation event is nondescript. But we have some reasons to think that it may actually be regulated depending on whether the cell is triggered by various conditions, such as perhaps viral infection. Now, to really investigate how this E3 sumo ligase domain. So this is an enzyme. It catalyzes this reaction, the transfer of sumo. And we want to know really how that works and how that modulates the function of RanBP2. So my lab back in 2021 developed a protocol that we could use CRISPR-Cas9 to edit the RanBP2 gene of tissue culture cells so that we could disable this E3 sumo ligase domain. And that's really key because if you want to understand how something works, first you have to start dismantling it and taking it apart. And so this is our attempt as to understand what this E3 domain was doing. And so we targeted a region of the RanBP2 gene by CRISPR-Cas9, and you can cut it right in the middle of exon 21, where the main catalytic region of the E3 sumo ligase is. And when you cut the gene, the cell repairs it, and it usually introduces mutations. So this is a particular cell line that we developed called RanBP2 dead E3, dead E3. And we have two copies of every gene. So in this cell line, the two copies of RanBP2 are shorter than the wild type if we amplify them. And they're shorter because one copy suffered a 45 base pair deletion and the other one suffered a much longer deletion. So one deletes a little bit of exon 21 and the other one deletes a little bit longer. And it results in defective versions of RanBP2 being produced. You can see that this cell line produces RanBP2, but if we look at the amount of sumolation, so this is RanGap1 and this is the sumo version of RanGap1, you could see that this cell line is defective in sumolation. So using the cell line, we've been able to try to understand what roles this sumolation catalytic event has in cells. Okay, and so I'm just going to go and hit a couple of things that we've discovered. And the first thing is STAT1 signaling. So when the body is infected by viruses or other types of infections, one thing that happens is that infected cells start to send out signals to warn the body that there's an infection underway. And one of these signals that's being released are type 1 interferons. And these type 1 interferons are signaling molecules which will bind to the outside membrane of a cell and precipitate a signaling cascade that ends up activating a protein called JAK1. And what JAK1 will do is transfer a phosphate onto STAT1. And the STAT1 then enters the nucleus and actually directly activates genes that are involved in the innate immune response. So this is the first line of defense against viruses. This is before the adaptive immunity kicks in, where you have antibodies that will target this virus. This is basically a signal saying I'm infected with something that I don't have necessarily antibodies to. So that's why it's called the innate immune response as opposed to the adaptive response. And so what we discovered is that RanBP2 tends to add sumo to STAT1. And this sumolation event of STAT1 prevents JAK1 from adding a phosphate and prevents STAT1 from going into genes. So what RanBP2 is doing here is temping down the innate immune response by preventing STAT1 from actually entering the nucleus. So we published this last year, and we're undergoing several types of experiments in the lab to see if this is related to other processes that we can see that are happening after viral infection. A second thing that we discovered is that RanBP2 tends to influence the formation of stress granules. And so what are stress granules? Well, typically when genes are activated and mRNAs are exported out of the nucleus to the cytoplasm, they're used to synthesize new proteins. When cells are infected with viruses, one of the things that commonly happens is that these viruses have an RNA genome, and that RNA has to be duplicated, or the genome itself may be double-stranded. And double-stranded RNA is kind of a signal that a virus is inside your cells. And this is recognized by several innate immune response modulators, and one of these is PKR, protein kinase R. And what protein kinase R will do then is just shut down all protein synthesis. It's a precautionary measure. And as a result, now all of these RNAs are no longer being used to make proteins, and these silent RNAs tend to accumulate in these cytoplasmic aggregates called stress granules. And these stress granules not only have silent RNAs, but they have these stress granule matrix proteins, G3BP1 and G3BP2. Well, a graduate student in my lab very recently discovered that. So the other thing, before I get to what Eva discovered, is that you can actually just trigger these by subjecting the cells to oxidative stress. And that will also turn off the translation of mRNAs into proteins and trigger stress granule formation. And what my graduate student found was that G3BP2 is also sumolated. So this is one of these stress granule matrix proteins. And this sumolation, what it ends up doing is it helps to disassemble the stress granules. And as a result of this, in the presence of active RanBP2, the formation of stress granules is a little bit tempered, and you more easily make protein. So this kind of tempers this inactivation of mRNA translation. And in a way, you would expect that it would dampen this innate immune response in response to viral infection, although we haven't tested whether the simulation of RanBP2 plays a role during viral infection. The third thing that my lab has documented is that RanBP2 seems to be intimately tied with the activation of the interleukin-6 gene. So interleukin-6 is a cytokine, and is one of the main cytokines that's associated with ANE. And so it seems that this happens in multiple ways. So one of the things that we have documented, and Natalie's lab has also seen, is that when in the presence of RanBP2, there's some sort of signal that feeds back to the IL-6 gene that prevents the IL-6 gene from making too much mRNA. We're not quite sure what the signal is, and it could be STAT1, but it could be other signals. But we're pretty confident that in the absence of RanBP2, the interleukin-6 gene is not as active. And when RanBP2 is disabled, interleukin-6 gene activation and the production of interleukin-6 mRNA goes up. In addition to that, my lab has documented that the interleukin-6 gene does something really interesting. It recruits silencing factors in the nucleus, and this is mainly through this protein called Argonaute-1 that binds a little tiny RNA that allows Argonaute-1 to find the interleukin-6 mRNA and bind it in the nucleus. And what we found is that once this entire complex is exported on the cytoplasmic face of the nuclear pore, it appears that RanBP2 sumolates Argonauts. And so what this ends up doing is it enhances the interleukin-6 mRNA silencing, and it prevents interleukin-6 from being translated into protein. And what we found is that if you don't have RanBP2, or if you disable RanBP2 using those mutant cells where we disable the sumo ligase domain, instead of being sumolated on the nuclear pore, Argonauts get ubiquitinated and essentially get ejected from the mRNA. And as a result, now the interleukin-6 mRNA is no longer silenced, and you have hyperproduction of interleukin-6 protein. And that's very interesting because one of the hallmark signatures of acute necrotizing encephalopathy is cytokine storms. And this actually directly implicates RanBP2 in tempering down the translation of interleukin-6 mRNA into interleukin-6 protein. Okay, now the last story that I have. So everything that I've shown you so far has either been published or has been submitted for publication. But I just wanted to share with you some really exciting new data that we have on another protein called TRIM5 alpha. And so this all started when my graduate student, Eva, in collaboration with another lab at University of Toronto, decided to investigate what proteins bind to not only RanBP2, but mutant forms of RanBP2 that are associated with ANEmone. And so the way we do this is we overexpress the N-terminal fragment of RanBP2 coupled to a couple of tags, and we either express the normal version or the version, or we actually combine 3 ANE1 mutation. So it's important to note that all documented mutations of ANE have only a single mutation, but we really wanted to make sure that we could identify proteins in cells that may recognize these mutations or that these mutations may disrupt. And so we incorporated actually three mutations together, and we express these in cells. And then what we can do is we can isolate each one of these proteins independently and analyze all the binding factors by mass spectrometry. So the idea is that RanBP2 is associating with other critical functional molecules in the cell, and perhaps these ANE mutations alter those protein-protein interactions. Typically, RanBP2 will associate with RNAs and other proteins, but here we're more interested in the proteins. Okay, so we did mass spectrometry. Now I'm going to show you some very complicated graphs. Don't worry, all will be explained. So this is just looking for proteins that associate either with the wild type RanBP2 fragment or the mutant fragment. And this is just comparing them to a control precipitate. This is actually looking at how statistically confident we are that these are real interactors. You can see that we pull down parts of the RanBP2 complex. So this is like RanGap and UBC9 and RAN, things that we know bind to RanBP2. We actually see lots of nuclear pore component factors, including parts of the Y complex and nuclear transport receptors. But we also see some additional proteins of interest. And if we focus on how these proteins interact with either the wild type fragment of RanBP2 or the mutant fragment, you could see that most of them fall on this line, saying that they interact both with the wild and mutant versions identical. But there are some proteins that either interact more with the wild type. So these are proteins whose association with RanBP2 may be disrupted by the ANE mutants. And actually, we find quite a few proteins that interact with the mutant form of RanBP2, but not the wild type, including some kinases such as CAM kinase. We have various CAM kinases that associate here. We have an E3 ubiquitin ligase, UE1, that also seems to be promising. But one of the most interesting proteins that we identified was TRIM5 alpha. So TRIM5 alpha is extremely interesting because previously TRIM5 alpha has been linked to viral infections. So it seems to be a factor that the cells make that's called a restriction factor that actively inhibits the reproduction of viruses. Now, in the case of TRIM5 alpha, its best documented activity is actually against the HIV virus. So it appears that when HIV viruses enter cells, TRIM5 alpha forms these purple cages around the virion, and that prevents HIV from rapidly replicating inside of cells. And interestingly, Natalie's group had previously documented that TRIM5 alpha is simulated by RanBP2. So this is doubly interesting because we can see that it associates with the mutant forms, and Natalie's group has found that actually RanBP2 simulates TRIM5 alpha. So does TRIM5 alpha interact with RanBP2? We wanted to confirm this not only with the triple mutant, but individual ANE mutants. And sure enough, so here I'm just showing you a blot where we immunoprecipitate either the wild type fragment of RanBP2. This is the fragment that has the three mutations. And now we have individual mutations, including T585M. And so we can immunoprecipitate our fragments down. And when we look to see if TRIM5 alpha is there, you can see that the wild type binds a little bit of TRIM5 alpha. The triple ANE mutant binds a lot, and single mutants bind significant amounts of TRIM5 alpha over the wild type. This is just a control protein. So it looks like the mutants, at least the three that we've checked, all share in common that they have an enhanced association with TRIM5 alpha. And so we live in the day of artificial intelligence, and maybe some of you have heard of this AlphaFold. So AlphaFold is a program that predicts protein structures. And so what we did is we inputted in the RanBP2 N-terminal fragment and TRIM5 alpha. And what we found is that actually AlphaFold predicts that these two fragments will only bind in the presence of mutants. And so in the presence of mutants, so this fragment of RanBP2 is in blue and TRIM5 alpha is in pink. And you can actually see right here that TRIM5 alpha actually inserts these two strands right in the middle of RanBP2. Okay, and if you zoom into where TRIM5 alpha is inserting these domains, actually it's right around where all the three mutants are. So it makes perfect sense that TRIM5 alpha would bind to these mutants more efficiently than the wild type. And in fact, when we stick in the wild type RanBP2 into AlphaFold, it doesn't predict this interaction. So this is in line with our biochemical analysis. And so, here we have this protein that binds to RanBP2, and it binds to this N-terminal region of RanBP2, which is involved in RanBP2 anchoring to the nuclear pore. And so we decided to ask whether the interaction of TRIM5 alpha displaced RanBP2 from the pore by basically occupying this N-terminal region. And this is exactly what we found. So here what we're doing is we're expressing a GFP RanBP2 without the mutations or with the ANE mutations. If we just express some random protein, you could see that the wild type and the mutant forms localize around the nuclear rim, so they're at the nuclear pore. And if we express now TRIM5 alpha, in the context of the wild type, you can see that there's not much difference. This cell that expresses the TRIM5 alpha construct looks like its neighboring cells. But in the case of the mutant form, when we express TRIM5 alpha, you could see that it displaces actually RanBP2 from the nuclear pore, and that's unlike a neighboring cell that's not expressing TRIM5 alpha. So it looks like, at least in the context of overexpressing TRIM5 alpha, it can actually displace the mutant form of RanBP2 off the pore. And so we're still working on TRIM5 alpha. I don't know that we have all of and the total insight of what RanBP2 and TRIM5 alpha association is all about, but it does look like TRIM5 alpha is a protein that has enhanced binding to these ANE mutant forms of RanBP2. And so, RanBP2 ends up having a lot of interactions with the innate immune signaling complexes, be it through STAT1 and inactivating interferon-stimulated genes, or whether it's AGO1, which disrupts interleukin-6, and we have some data that this is also true for TNF-alpha mRNA translation, and it seems to also affect stress granules. And in the context of ANE mutations, there's this interaction with TRIM5 alpha. We don't know yet what the effects, and we don't yet really understand how simulation may impinge on this unknown function, if there is one, between TRIM5 alpha and RanBP2, when at least it comes to viruses such as influenza. In the context of HIV, we know that simulation, this is nice work by Natalie's lab. It seems like TRIM5 alpha simulation diminishes its ability to prevent downstream consequences from HIV. Okay, with that, I'm just going to thank the people in the lab who did the work. A lot of what I presented was the work of Eva Wang, who's up here, with the help of a new student that I recruited recently, Aisha Desjardins. And there's some help from Sonu, who is right here. And I'd really like to point out my former postdoc, Qintang Sheng, who has a position now at Fujian Medical University in China. Okay, and with that, I'm going to end my presentation. Maybe Natalie, be great if you put the volume on.
Sorry, you missed all that chatting. You're
Great. Do you want to take questions now, or are we going to wait till later?
Yeah, we have some questions. I'm just trying to see how we do it. Alex, can you hear me?
Yes.
Perfect. Hi, Alex. Sorry you're not here. It would have been nice to have you here to answer these questions and give that talk. That was a great talk. I have a question regarding cell types and specificity, and you know, I'm going to
Yes.
ask a neurologist. I think about nerve cells and other things. So do you think that is there differential expression of RanBP2 or placement of RanBP2 depending on the cell type within the body? Or is this a general, these are general operating rules for every cell in the body? And you understand where I'm coming from, right?
Yes. 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 So as far as we understand, RanBP2 is ubiquitously expressed, and that for the non-scientist, that means that it is expressed in every cell of the body. And
So can I
as
follow
far
up?
as, sorry?
I was just going to ask a follow-up question, but you're probably going to answer it right now.
Yeah, so it is expressed in every cell in the body. As far as anyone has looked, it is always on the nuclear pore. But in addition to being on the nuclear pore, many groups have seen that there are cytoplasmic foci of RanBP2. And this is a very contentious point because there are some groups out there that think that the um, non nuclear pore complex associated RanBP2 may play a role in cellular functions. Jamon Joseph, who's at the NCIC at Pune University in India, has documented that there are cytoplasmic foci of RanBP2 that he thinks may associate with mitochondria. I know that Paulo Ferreira had documented quite a while back that there are huge cytoplasmic puncta of RanBP2 in retinal cells. And I have to say that depending on the cell type, we can see various amounts of the cytoplasmic puncta of RanBP2. And we're not quite sure what those do and if they play a role, and if those are, you know, found in some cell types rather than others. I have to also say that there is unpublished work from Rick Wozniak's lab, although we have yet to confirm this, that in some cell types, if you stimulate them with cytokines, you could see a massive upregulation of RanBP2, and they tend to accumulate in these cytoplasmic foci. Now, we haven't really seen this, and it could be that this is a cell type dependent event. The other thing to think about in terms of RanBP2, we do think that one of the critical things about RanBP2 are is its E3 sumo ligase activity. And it's very likely that enzymatic activity may be regulated in a cell type dependent manner. And it's very likely that depending on the cell type that you're in, RanBP2 may simulate different types of proteins. And it's also likely that that is subject to the regulations of the innate immune signaling and other cellular signaling processes, which may vary between different cell types.
Thank you. That was really helpful. And I think that that sort of leads to the concept that in order to really understand this, if you start from the molecular level, like where you are, but then think at the organism level and ask the question of the differential effects of having a particular gene mutation or abnormality, and why that gets driven in different, you know, for us, the central nervous system almost exclusively.
Yeah, I agree. It's very puzzling. This is a protein that's expressed everywhere. It seems to play very intricate roles in modulating the innate immune system. And I think going forward as a community, it's going to be really important for us to determine what is the critical cell type. It is possible that it's going to end up being multiple cell types. Um, but I agree with you completely that it's going to be critical to understand, um, what are the relevant cell types in terms of our A&E patients. Um, and not only the relevant cell types, but the relevant conditions. I mean, a lot of the experiments that we've been doing are in cells that are not virally infected. We can see that if we start triggering cells with immune signals that mimic a viral infection, a systemic viral infection, RanBP2 is modulating this. So it could be that RanBP2's major function is actually not in the virally infected cells, but rather in secondary cells that are sensing signals of viral infection. And that's possible.
Hi, this is Vanessa from Imperial. Thank you. Really enjoyed your talk. I work on inborn errors of immunity. And I just had a thought. You described you see cytoplasmic puncta around BP2. Could it be possible that just because we see similar things with herpes encephalitis, that autophagy could be involved in clearing out? Could RanBP2 be targeted by autophagy for clearance, and what you're seeing in the puncta is actually sort of destruction in the autophagy mechanism, and that's causing sort of this inflammatory response somehow?
It is possible that those cytoplasmic puncta could be related to autophagy. Um, we have tried to identify the nature of those cytoplasmic puncta. As I pointed out, there's a lot of debate in the field as to exactly what those are. It appears that a subset of them are associated with the endoplasmic reticulum, which would be in line with them being autophagosomes. Although I think most people in the field would say that these are actually this structure called annula lamellae. Annula lamellae are reserves of nuclear pore proteins that appear in the endoplasmic reticulum in rapidly dividing cells. Um, but I have to be honest with you. I think that we don't really understand what those puncta are, and it could be that they're autophagosomes. It's entirely possible.